Welcome to Spring LIB.
Hello students, the question says that in 1953 Watson and Crick published a paper in the magazine Nature about the molecular structure of nucleic acids. At the end of the paper they claimed that it is not escaped our attention that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. Explain what it means by describing the structure of nucleic acid and the mechanism of DNA replication and RNA synthesis. So we have to explain the nucleic acid structure with regard to the above statement, also replication and transcription process. Coming to the nucleic acid structure, as we know the nucleic acid as it could be DNA or RNA, which compose of long chains of nucleotides, and each nucleotide consists of three components: sugar molecule and phosphate group and a nitrogenous base. And in terms of nitrogenous bases, there are four types of nitrogenous bases: adenine, guanine, cytosine, uracil. Cytosine and thymine. Uracil is found in RNA instead of thymine, which is found in DNA. Now coming especially to the DNA, DNA has a double helix which is formed by the base pairing of A with T and G with C, which corresponds to the above statement that specific pairing we have postulated immediately suggests possible copying mechanism. So this base pairing is responsible for the double helix of the DNA structure, and these bases are held together by hydrogen bonding. And these base pairs on hydrogen bonding are crucial for DNA replication as well as transcription process. So DNA double helix is because of the base pairing of A with T and G with C, which are held together by hydrogen bonding. Coming to the DNA replication, as we know that DNA is double helix, and before the replication, which means that the copying of the DNA is formed, this DNA is unwound first, then new copies of the DNA strands are synthesized, forming the double-stranded two molecules of DNA. And these new strands are synthesized mainly because of base pairing mechanism, in which A pairs with T and G pairs with C. So this replication process would not have been possible if there was no base pairing mechanism in place. So in DNA replication, after the DNA is unwound, both the strands they act as templates for the synthesis of new strands, forming a double-stranded two molecule base pairing mechanism. Now coming to the transcription, in which RNA is synthesized from DNA. As already said that DNA is double-stranded, this is unwound and only one strand acts as a template to form mRNA molecule. So the DNA is unwound and only one strand forms the template for the synthesis of mRNA molecule, which is synthesized by RNA polymerase. And this RNA molecule, after the synthesis from the DNA template, then form the proteins. And this mRNA is also formed due to base pairing of A with T and G with C, which form hydrogen bonding. And as we know that in each and every case, there are two hydrogen bonds between AT and three hydrogen bonds between GC here, and which are responsible for replication as well as transcription process. I hope you found the video useful. Thank you.